Here. Okay, so for this course students, I want to remind you that if you press on the next button here, you will get to this next lecture page. And you always have the PDF that is uploaded. We upload the PDF the day before. Now it's a little bit slug because of my Zoom, I think. But anyway, you can get the PDF from here or download it, so you can write your notes if you want. And then every day we will put the recording of the lecture here, the link. And for people who don't have access to Canvas, people online, we upload the recording of the lectures every evening to our YouTube channel. You can find the link on our website. Okay, so now we're here, and this is the basic optics for light microscopy. So that's what I'm going to talk about now. Right. So I think it's important to understand a little bit how an image is formed so that you can understand the consequences of you being a bit sloppy with your sample, maybe, and also the importance of the objective and all this type of things that we will talk about later during the week. So let's do a little bit of optics. This stuff is not going away here now. So when light, as I said before, when light travels in a heterogeneous manner, it may encounter a new medium or an obstacle, right? And this is what happens when the light goes through our sample, it hits the cover slip, whatever, it goes through the mounting medium, etc. So here, so 4 things can happen. Reflection, refraction, diffraction, and interference. So we're going to look at them now and see what they mean. So first, let's talk about reflection. So this is what happens when the light hits the boundary between 2 different media and the light bounces off without continuing into the new medium. So you can think this is the boundary. Here we have one medium and here we have another medium. And these are 2 different media, and the light comes up here with a certain angle, and then it doesn't go through the other medium, it just bounces off, okay? So this is what happens with this cute little cat, and we can see its image. So it's the light that comes from the cat has been bounced off the mirror. So what happens is the light changes direction, but the shape and the speed of the wavefront are not changed. So you remember, this is ray optics, and this is the representation of light with wavefronts. And so these are not changed as long as the boundary is flat. So the light will come here, and then it continues with a different direction. So how is this relevant for us? Well, in the microscopes, we have many different types of mirrors, and we use them to direct light to wherever we want in the microscope. There are lots of right angle turns in the microscope, so we need the mirror to direct the light wherever we want. And also, this is an image that you can take if you find a beautiful dress in a shop window during the night, and you say, oh, I'm going to take a picture of this dress with my flash, and I will send it to my friend. And this is exactly what happens. So the light from the flash bounces off the window, and there's no way you can send that to your friend. So this is reflection. And this happens also in our samples because we have this very strong excitation light that we are sending to the sample, and it, hits the cover slip, for example. And a lot of light is reflected on the cover slip and goes back into the objective. We clearly do not want to image this excitation light. And this is the reason why we need dichroic mirrors. So we are going to talk about this tomorrow. And so you will know everything about dichroic mirrors very soon. But basically, this is why this is relevant for us as microscopists, because we use mirrors to direct the light, and we also want to get rid of the excitation light that reflects of the sample. Now, if we look at refraction, this is the second thing that can happen when light reaches a boundary. So it's also the boundary between 2 different media. So here we have one medium, here we have another medium, and here is the boundary. But this time the light goes through the boundary, so it enters the second medium. And the light is transformed by this, the fact that we have two different media. And you can see this, it's when you're a kid and you want to impress your parents or something, or you discover that you think it's a lot of fun. So you put a pencil inside a glass of water and say, look, my pencil is broken. This is what happened when you have refraction. So light has been transformed in such a way that it changes speed. So you can see here the wave fronts, they get tighter. So it changes speed, it changes direction if the light doesn't go straight through. So if it's perpendicular to the boundary, it will keep going without changing direction. But if it's at any angle, it will change direction. And this is why this stuff looks broken. It changes direction, and it also changes the wavefront of the light is actually not changed if the boundary is flat. Like here you see this is a flat wavefront, and this is also a flat wavefront. So the shape is not changed if the boundary is flat. So because of this, there is different materials have different properties for how they refract light. And so there is something called the refractive index of a medium, and this is often or always abbreviated as a small n. So when you see a small n like this, you will know it's the refractive index. And this is a measure of how much the medium changes the speed and the direction of light compared to air or vacuum. So air and vacuum are the reference, and therefore they have a refractive index of 1. But glass, for example, has a refractive index of 1.52, depends on the type of glass, but more or less around there. Water, PBS, cell culture medium, and cytoplasm in many cells have a refractive index of 1.33. And we will be working with these numbers quite a lot. You don't need to remember them because you can find them all over the place on the internet. So no need to remember. You will anyway remember because we're going to be using them a lot. So, and there is a law. So it's a very simple law, if you want to say, Snell's law that regulates how much the light is changed based on the difference in refraction index from the first medium to the other one. So we have, this is the angle. So the two angles are related to the difference in refractive index. And different wavelengths refract differently. And that is, in some ways, an advantage for us because we can make a rainbow. So we can separate the different wavelengths that are in white light. This is air, so it has a refractive index of 1. And here is a prism, a glass prism. Sorry, so the refractive index is much higher. And so what happens here is that we have the light comes at an angle. And so we have this light is being dispersed in its different wavelengths. And when it comes out, it again changes medium. And then it's being dispersed even more. And so we are able to make a rainbow. And that is very relevant for microscopy because we use this in spectral detectors, either using a glass prism or using a grid, which has the same effect. But basically, we're using the fact that light can refract so basically, to disperse the light and see the different colors separately, then we can image them separately. So very useful for microscopy. But also, samples are made of heterogeneous matter. And so, when you have lipids in your sample, you have membranes, maybe you have cytoplasm, and you have proteins on the outside of the cells in the extracellular matrix. All these have different refractive indices. And so light refracts at each refractive index boundary. So that's a lot of refraction in our samples that happens. And then every time we are getting all this spreading of the light all over the place. So when we do sample preparation, when we prepare a sample, it's important to place the tissue on the cover slip or the cells on the cover slip directly, not on the slide, if we can. I know it's not always possible because sometimes we get samples that are old and they have been prepared on the slide. Sometimes we get samples that are cut at the hospital and then they put them on the slide. They don't want to put them on the cover slip. Sometimes we have samples that may detach, but if we can, we definitely should put our samples on the cover slip, not on the slide. We will get much better images. And so we will talk about all this later. But the reason why we are getting worse images when it's on the slide is because of the mounting medium. And we have refraction in the mounting medium. So it also has an influence on the objective, the choice of objective that we have. We should match the medium that we use on the objective. For example, we use an oil objective, we're going to put oil on the objective, or we use a water objective, we're going to put water. That medium has a certain refractive index, and we should use the same refractive index as the sample. So if we have a sample that is quite fatty, maybe we want to use an oil objective. If we have a sample like live cell, so just cells in culture, then we want to use a water objective, etc. And that is also to avoid distortion because of refraction. So we try to minimize refraction. Inside the microscope, light travels in air, right, inside the microscope, but once in a while it finds glass lenses. And there we have refraction exactly in the way I've shown in here. So we can use lenses to manipulate light. And I'm going to tell you this a little bit later. And that is also very useful. So refraction in some ways is negative for us, and in some ways it's positive. And we can use it to image our sample in a better way. And then the third behavior of light when it hits a boundary is diffraction. So let's see what that is. So now we have the same medium. So in the other one, in reflection and refraction, we had two different media, but now we have only one medium, but there is an obstacle. And this is a solid obstacle the light cannot go through. So the light will travel and it hits the edge of the obstacle and it continues. So it is influenced by the obstacle, and this is what diffraction is. So what happens is that the wavefront is bent around the edge of the obstacle. So if you look at this very nice and inviting beach, you can see that we have waves coming in here. You see, they are a little bit parallel. So we can see here, for example, some pattern, and we can see here some pattern, and we can also see here a different pattern. So in here, we have a flat wavefront, and away from the pier, the wavefront continues flat, right? I can show you on and off so that you can appreciate that it is flat in here. However, near the pier, the waves are bending, and so we get a curved wavefront. Exactly the same thing happens with the light when it hits a hard boundary that it cannot go through, but that has an opening. So it can continue straight or it can bend around, but it does this in 3 dimensions. Of course, on the sea, it's just 2 dimensions, but for us, we have it in 3 dimensions. So what is the relevance for us microscopists? Where every molecule in the sample is an obstacle and it diffracts light. And the edges of the objective, you can see this as one of the side of the objective, is a hard boundary that the light cannot go through. And then there is an opening. So we have air at the outside. For example, if it's an air objective, we have air here, and then we have air inside, and then the light will continue through, but at the edge, it will bend. So the edges of the objective are hard boundary, and so we have diffraction there. And actually, we will see that in the next little lecture. The image formation comes from the diffraction and the interference of light in 3 dimensions. So diffraction is a very important phenomenon. Without diffraction, we have no image, so we cannot do any microscopy. But here there is another word called interference, and this is the 4th behavior that I was talking about. So let's see what interference is. Interference is actually can be a consequence of diffraction. It's basically, we have here this flat wavefront. Again, it's the sea. And then in here, we have an obstacle. Some of the light is going to continue straight and still have a flat wavefront here. But much of the light is going to be diffracted off these 2 hard obstacles in here. And so it will bend, like we have said before. So we have diffraction. But what happens when these waves are coming in here and they are diffracting, and they will be bent this way, whereas this wave are diffracted in the other direction. Here in the middle, they are meeting. And if they meet, there is interference. So we have interference here where these waves and these waves meet, and we have interference here where these waves and these waves meet. And if you like to kayak, you will see that even if you have a pretty chirpy sea, you may find a little pool of quietness behind an island. So this is because of interference. So waves that are traveling in one medium, but coming from different directions, they bump into each other. And I'm sure you remember this from your childhood class, that the waves, depending on if they are in phase or out of phase, so these ones are out of phase, this one goes up when this one goes down, then they will interact and interfere in either constructive way when the wave becomes higher, or in a destructive way where the wave becomes bigger. So we are getting basically an interference pattern out of these places where we have interference, and the amplitudes add or subtract. So how does that affect us as microscopists? Well, light is a very, very regular wave. And so it's very unlike the sea, where you're influenced by the wind, for example. The interference pattern is highly predictable. And so that is to our advantage. The interference pattern carries information about the wave, but it also carries information about the obstacle, because the obstacle is the one that creates these diffraction in here. And so we can get information about the sample thanks to interference here. So when we record an image, microscopy in it, what we record is actually the 3D interference pattern from all the diffraction events and all these waves bumping each other in 3 dimensions. This is what we record. So anything that affects the interference pattern in our optical train, so all the way from the light source, which in our case in fluorescence microscopy is the sample itself, is the fluorophores. So from the fluorophore all the way to the sensor, anything that affects the interference pattern will also affect the image. So now I would like to, I have just mentioned a little bit that we have diffraction and we have refraction in lenses. And so we can use lenses to manipulate light, and we can also use irises. So let's have a look at how we manipulate light using these 2 types of objects. So first, lenses. The light will change direction, speed, and wavefront shape. So wait a second, I told you that the wavefront shape was not changing when we have refraction, but this is only if the boundary is flat. So if the boundary is flat, we have flat wavefront here, flat wavefront here. But as you know, lenses are not flat, or they can have one flat side, but they never have 2 flat sides. So here we have all sorts of different lenses, and you can see here in objectives, like the most complex objectives, you have lots of lenses that are combined. So here is a lens that have a curved side and a flat side, and you can see here in ray optics, the light that comes in. The light that comes in perpendicular to the surface it keeps going straight. So it's not bent by refraction. But the light that comes in on the side is going to be bent when it comes into the lens, and bent again when it comes out of the lens, okay? Because of this, we can have a focal point. So in here, what we have is the lens is made of glass, so it has a refractive index of 1.52, and the air is 1. So this is an air objective. So we have refraction. And here we have a flat wavefront. And because of this refraction at all the spaces except in the middle, we have a focal point. All the rays here are going to focus on one point, and here we have a curved wavefront. So this is the way we can focus light that is defocused. We can manipulate it with lenses and make it focused. And we can do the opposite as well. We can use some light that is focused and make it not focused. So here, I'm sure you remember this also from your school time, where we have a little object. Sometimes it's like shown as a candle. And then we have a lens, and the light that goes through the center is going to continue straight, and the light that goes to the side is going to be refractive, and then it focused here. And so all the light that comes in here is going to be focused here, and vice versa. So we're getting here an image. We're getting an image of the sample, which is inverted, and then we have another lens, which is going to put that thing straight and maybe magnify it a little bit. And here we have another image. So here we have an image and it's the final image plane. This we have an image and it's the intermediate image plane. And here we have no image in between. Light is traveling, but there's no image. So we have definite planes where the images form. So in our microscope, we have a point light source. This is our fluorophores. They are like a little lamp inside the microscope, and so they are inside the sample, and so they are going to shine light when they're excited. They're going to shine light in all dimensions of space. But we have the objective here, and some of that light is going to enter the objective, and the rest is going to be lost. And here we have a refraction, and so the wavefront is going to change. Here it's a curved wavefront, here is a flat wavefront. And then we have another lens, and we're going to focus this light in and have an image. And so this is the intermediate image plane exactly that we have up here. Then we put more lenses, etc., and then we end up with something that is called the tube lens. That's the final lens that we use in the microscope, and it is going to refocus the light at the final image plane, and this is where we put our sensor. So we're going to record this. So in microscopes, refraction through lenses is very useful. There are some spaces where the images are formed, and these are called image planes. So here is an image plane, and here is another image plane. And the rest of the space, there's light traveling through, but there is no image. So this is the way we manipulate light using lenses. But we can also manipulate the light using irises, and this is what they look like. It's a little diaphragm that can open and close. And depending on where they are in the along the light path, they can be called field stops or apertures. So here we have our sample with the objective and all the way to the tube lens, and then the sensor here, we record the image. So if we put an iris like this at an image plane, then it's called a field stop. So you can think that this is where the image would form. So you can remember field of view, field stop. So the place where we have the image is actually called a field stop. And what happens when we close this iris is that it's visible in the image, because this is where we have the image. So we're looking at the image, I close the iris, I can see it. So it works a little bit like a curtain. It blocks the light from the outside of the field of view and towards the center. So if you close it completely, it will be a very small hole there in the center. That's a field stop. So it only affects the edges of the image. The center of the image is exactly the same if the field stop is open or closed. If, however, I put the iris outside of the image planes, somewhere where there is no image formed, but the light is traveling, then it's called an aperture. And when I close the aperture, because there is no image there, I do not see this curtain effect. Instead, everything, because the light, we are preventing the light that is traveling here from participating in the final image, then the whole image is affected. So we prevent some light from participating to the interference pattern at the image planes. So we are affecting everything in here, not just the edges, okay? So this is the way apertures work. When you close them, the image will become dimmer, and so there's quite a lot, the quality of the image changes, whereas a field stop, it's only, it closes like a curtain. So how is that relevant for us? So we have field stop and apertures depending on where they are along the light path, and how is that relevant? Well, field stops are visible in the image. We can use them, for example, to prevent illumination outside the field of view. And this is used a lot in transmitted light microscopy, but it's also used in fluorescence microscopy, but maybe you do not see it because it's automatic on your microscope. Might or might not, it depends on what you do with the camera. But basically, if we want to image only this part of the sample, we don't want the rest to be illuminated, for example. And so because it's going to bleach and we're not imaging it anyway. So maybe we will want to close the field stop to be able to restrict the light to only the place that we want to image. This is one of the applications of a field stop in microscopy. And you will have to do this when we do the color illumination exercise. You will have to align your microscope for transmitted light microscopy, and then you will have to close a field stop. So it's used in the color illumination. We will do that later. Whereas apertures are not visible in the image, but they affect the whole image. And this is also used in transmitted light imaging to adjust the contrast. So you will also need to use an aperture and close an aperture when we do the color illumination exercise. And most importantly, the edge of the objective acts as an aperture. If you think about the edge of the objective, it is a hard boundary, and the light is going to travel through the objective, and it's going to bend on the edges of the hard boundary. And so the edge of the objective acts like an aperture. And this is why there is a very important specifications of objective called numerical aperture. Okay, so an objective that has a small numerical aperture, and again, we will talk about all this when we talk about objectives, I think in 2 days. When an objective has a small numerical aperture, it means that the aperture is closed. Okay, so we're more in this situation than in this situation. So less light is involved in building the interfering patterns at the image plane. So the image is dimmer and the resolution is lower. So the quality of this image is different from this one. So this is how this affects us as microscopists. So in conclusion, in a microscope, light encounters different media and obstacles, and that leads to reflection, refraction, diffraction, and interference. And we can manipulate light by using lenses. So we can change the light direction, the speed, and the wavefront. We can focus the light at image planes. And we can also use irises, which are these small diaphragms. And they can be called field stops or aperture, depending on where they are. If they are a field stop, when we close them, they work like a curtain. And if they are apertures, then they affect the quality of the whole image. So now I'm going to go into another little lecture called image formation. But before I do that, I just want to check the schedule to see if I'm